Armando Hasudungan, biology and medicine videos. Please make sure to subscribe, join the forum group. For the latest videos, visit Facebook Armando Hasudungan. Here, please like, and also you can ask questions, answer questions, and post some interesting things, including your artworks. And you can change the quality settings to the highest one for better graphics of these videos. This video is going to be on membrane transport and the different forms, types. So, membrane transport, it's important that the cell transports water in and out to maintain osmolarity. Also to transport glucose, for example, inside to utilize energy and to make ATP. But how do these molecules move in? Well, membrane transports help them move in, you can say. And there are a few types of membrane transport. There's passive diffusion, there's facilitated diffusion, there's active transport, where we can have primary active transport, secondary active transport, and translocation. And then we have just the fourth one, bulk transport. The question is, why do we need these different types of membrane transporters? Why can't the molecules itself just pass through? Well, actually, there's some molecules that can just diffuse through without any help or facilitated help of membrane proteins. So for example, if we have the extracellular fluid here, there are hydrophobic molecules, and these are things such as oxygen, carbon dioxide, and nitrogen. And they can easily diffuse through via diffusion without the assistance of membrane transporters, membrane proteins. Then we have small uncharged polar molecules, and these are things such as water, urea, and glycerol. And they too can diffuse through the membrane lipid bilayer without the help of membrane transporters. However, when we go to large uncharged polar molecules such as glucose, it's large and too polar to pass through, and so it can't, and therefore requires a transporter, a protein membrane transporter. And it's necessary for glucose to go through the cell because glucose is necessary for glycolysis and therefore to produce ATP for energy. Similarly, we have ions such as cations and anions, sodium, potassium, calcium, and we have chloride, negative chloride also, that cannot pass through because of its charge, and so requires the help of membrane transporters to go through. This scale I'm drawing shows how permeable molecules are, the top being highly permeable molecules to low permeable molecules. And these numbers I'm drawing, they are permeability coefficients, which is how many centimeters a second a molecule can move through the lipid bilayer. So for example, if a molecule was highly permeable, 10 to the negative 2, it can move 10 to the negative 2 centimeters a second through the lipid membrane, which is very fast considering a membrane is in nanometers, not centimeters. So centimeters is pretty massive. If the molecule has low permeability, such as 10 to the negative 14, that is so slow. That's 10 to the negative 14 centimeters a second. Water moves at about 10 to the negative 2 centimeters a second, and that is why it can diffuse without any facilitated help through the lipid bilayer. Glycerol, similarly, is about 10 to the negative 6, 10 to the negative 5 centimeters a second, which is still pretty fast. Glucose molecules move very slow now at about 10 to the negative 7, 10 to the negative 8 centimeters a second, and so they would require a membrane transporter to go through. Potassium and sodium at about 10 to the negative 11, 10 to the negative 12 centimeters a second are very, very slow, and that is why we always see ions moving through the membrane bilayer via membrane transporters. Now, assuming a lipid membrane bilayer is 10 nanometers across, this would mean that it is 10 times 10 to the negative 7 centimeters across, which simply means it is 10 to the negative 6 centimeters. And using this amount, we can measure how long it takes in seconds for each of these molecules to go through a 10 nanometer length membrane. So for example, for water, it takes 10 to the negative 4 seconds to pass through a 10 nanometer lipid membrane, which is very fast and spontaneous. Similarly, glycerol takes only 1 second to pass through a 10 nanometer membrane. However, potassium and sodium, these ions, take 10 to the 6 seconds to move through a 10 nanometer membrane, which is equivalent to about 280 hours it takes to move through a 10 nanometer membrane, which is very, very slow. But essentially, or theoretically, these ions actually do move at a rate through the lipid membrane bilayer. So these actually go through, but very, very slowly. And so we can just safely say that they don't move at all and that they always require a membrane protein transporter. Going back to the different types of membrane transporters over here, we'll just go through a brief run through at the different types we have. So here we have the lipid bilayer and the extracellular fluid and intracellular fluid. Now the first type of membrane transporter is the passive transporter. And this is essentially diffusion of a substance across the cell membrane, you can say. It can be with the help of a membrane with an actual protein or not, because some of these molecules, such as oxygen, can pass through quite easily. And then we have number 2, facilitated diffusion, which is highly specific, and it requires the help of proteins. And these ions or molecules that move through actually transport down its concentration gradient. So for example, here we have a channel, protein channel, and then we have high concentrations of protons, hydrogens in the extracellular fluid. And so simply enough, this channel protein will assist hydrogens to move down into the intracellular fluid, down its concentration gradient. And then we have another type of protein transporter, which once a proton binds to it, it makes a conformational change, so switches, therefore releasing the hydrogen in the intracellular fluid. And this transporter is known as a carrier protein, which is a part of the facilitated diffusion. Then we have active transport, which uses energy to move molecules through the membrane bilayer, and usually involves ATP. There is also electro and chemical gradients involved in this process. So typically ions are involved. So for example, we have a sodium in the extracellular fluid. It can bind into this special protein, where the protein will make a conformational change, basically flipping over, but this process requires ATP, as you can see, energy. And so sodium can be put into the intracellular fluid, and as you can see, it requires ATP. This protein is known as a pump. Another type of protein active transporter is a co-transporter, but this doesn't necessarily need ATP, and the movement usually involves two molecules. So for example, sodium and iodide can move simultaneously through the lipid bilayer by a co-transporter, and the movement is actually facilitated by one molecule's going down its concentration gradient to pump another molecule through. In this case, sodium gets pumped through to help iodide get pumped in. Many types of co-transporters as well. Next, we have bulk transport, which is essentially exocytosis of endocytosis. So for example, this is an example of endocytosis, where molecules get brought into the cell by the membrane forming a vesicle. And that concludes this video on membrane transports. However, I've only briefly talked about the different types of membrane transporters, and it might have been pretty confusing. So I've provided links to each of these different types of membrane transporters, and these other videos will talk more deeply into the different types: passive transport, facilitated diffusion, active transport, and bulk transport, specifically endocytosis. And it will also show you examples. So please click on the link provided in the video. Just click on it. Please like, comment, and subscribe. Thank you.